flying insects and flying spiders.
All right, thank you. So I threw in the spiders as another interesting case. And what I'd like to do is to give you some highlights of a lot of different projects that fall under this umbrella of flight and the smallest insects. And I am in the departments of biology and mathematics, but I've worked with mechanical engineers, physicists, oceanographers, biologists, and so forth. So a lot of the work will come from these various fields, and then I'll try to connect it together and talk about how we can use these interdisciplinary approaches to address some very challenging problems. And so our goal, looking at these very tiny insects, is to think about how the details of their morphology impact their performance as they disperse across landscapes and ultimately their success as a species. And there are many motivating questions. So one question is, if you take something like these super tiny insects that are anywhere from 300 microns to a millimeter, how far do they travel? Sometimes, presumably, they can travel hundreds to thousands of miles. I should think about kilometers. I'll say also hundreds to thousands of kilometers. But they're very hard to track because they're 300 micron specks within our atmosphere. And then what are the flight aerodynamics of tiny insects and spiders? So they operate at Reynolds numbers around 10 to 100. So the fluid dynamics is quite interesting. In addition to fluid dynamics, there might be other physics responsible for long-distance dispersal. So why do we care about what these insects do? And I'll include spiders here as well. So in terms of the flying insects, thrips are pests, also spider mites, so they cause significant agricultural damage every year, but they're also used in biological control. So parasitoid wasps and spiders are often released to control populations of pests. And there is a lot of work trying to find parasitoids that will control particularly invasive species in new areas. And I'll talk about some examples of that. They can transmit pollen, which is a good thing, but they can also transmit pathogens, and they can travel hundreds to thousands of kilometers. And I did update that for here. Okay. So how tiny is tiny? So I'm talking about insects that look like this fairy fly. This is 200 microns, and you can compare this to unicellular organisms, paramecium, amoebas. And despite being so small, they do have tens of thousands of neurons and a brain, all packed within this little tiny body. So there are also interesting questions from the physiological context about miniaturization. Now, working with Ty Hedrick at UNC, we were able to obtain some of the first videos of these insects in flight. So this is a thrips. And thrips is both plural and singular. And it is taking off from a pipette tip, and this is filmed at 4,000 frames a second. So they fly about 200 hertz. And essentially what you have to do is to take your high-speed video camera, and if you have more than one, focus them all on this side of takeoff, and then have an undergraduate student sit and wait for the animal to take off, and then you have a couple of wing beats that you can film. And the only footage that I know of that anyone has for these guys is takeoff. So you can imagine if you're just aiming your camera at a very small millimeter cubed area of space, and you're waiting for a thrips to fly through there, you would have to be very patient and spend quite a bit of time doing that. So we are trying to work at some technology to be able to film them in longer flight, but right now we just have the first few wing beats after takeoff. Now, spider ballooning, which is when the spiders fly on a single strand of silk, is even, I would say, less well studied. And this is a YouTube video. Really the best footage that you can find is from amateurs who, during these big ballooning events, happen to go out with their cameras, and you can see these little strands of silk flying through the air. So this was a big event in October 2012 where the skies were filled with ballooning spiders. So the conditions were just right for these guys to disperse quite long distances. But the silk threads are very hard to film in the lab, and I don't know of anyone who's done that. And hopefully, working with some collaborators in mechanical engineering, we'll be able to do that. So what we know about spider ballooning, a lot of it is anecdotal. So on one of Darwin's voyages in the Beagle, they were out at sea, 3,200 kilometers offshore. And one day, all these baby spiders just showed up on the Beagle. And so the thought was that these came from land, but they were very far from land. So they must have traveled that distance to get to the Beagle. They've also been observed at heights up to 5 kilometers. So pilots have seen baby spiders as they're ascending and descending. So these anecdotes are all we really know about how far they can travel. They use single or multiple threads, up to 100 milligram adults balloon. It's something you typically see in the juveniles, so they want to disperse to get away from all their brothers and sisters. But similar to thrips and these tiny insects, the wind speeds must be low for them to disperse. So they don't wait for a hurricane and allow very strong winds to take them far. This is done on sunny days with light winds. So one of the first projects I want to tell you about is trying to look at some of the measurements of how far any of these insects have dispersed, and then try to come up with some simple mathematical models to describe that behavior. And this was part of a year on mathematical and statistical ecology at the Statistics and Applied Mathematical Science Institute. And I was there with a few fluid dynamicists and a lot of ecologists who don't really think about fluid physics and mechanics. And our goal, more or less, was to convince them that they should think about fluid dynamics, particularly when it comes to dispersal. So typically, the models that they use is that they assume that diffusion or dispersal can be modeled as diffusion, and they do not consider necessarily what is happening in terms of winds or currents. And I guess I'm mostly talking about air here, but I think some of these rules could also apply to the ocean. All right, so one of the motivations here, how far can tiny insects go and other arthropods like these spiders? So it has been known for some time that these insects and spiders can travel long distances at altitudes that correspond to the types of tops of nighttime inversions, so wind and temperature inversions. And again, you can see this. Pilots have seen it as they're taking off and descending. So is there any more rigorous quantitative data that we can actually use to validate models of dispersal? And we probably spent the first 3 months looking at papers and contacting entomologists and trying to get someone to work with us. And we were very fortunate to find Nadia Christensen, who was in Australia at the time. And SAMSI provided us funding to fly her in and talk to her about what she had done. So she participated in this very unique study where it was the first time release of a non-native parasitoid in order to attempt to control whiteflies in Australia. And they released all of the parasitoids right here in this field. And they were interested in how far the insects could disperse, and they thought that their dispersal had been underestimated. So originally, they were only going to look at these nearby fields, but they decided that they would look for their emergence in a couple of farther sentinel fields, assuming they might not even show up there. And then they thought, well, maybe they should measure the wind speed as well. So it was really nice because you have a point release in space and time of these parasitoids. You know that if you find them exactly where and when they came from, you have people measuring wind speed. and some points in space. And so as far as we know, this is the best data that's out there. So unfortunately, the predominant wind speed is in this direction, and their measurements are taken in this direction. So they did not know the predominant direction when they set this up. Okay, so my postdoc, Christopher Strickland, came up with a drift diffusion model with some really, really simple assumptions just to see if we could describe their data. And we made some assumptions based on the biology of these parasitoids. So we assumed each wasp will undertake at most one windborne flight, that they move in the direction of the wind at a distance proportional to the wind speed. And because we only had one measurement of the wind at one point in space every 30 minutes, we assumed that the wind velocity was spatially constant in the study area, and it varied temporally according to collected data, which was sort of the best we could do given what we had. And the windborne flights had a fixed duration, so it was known that a similar species could fly in the wind tunnel for up to 30 minutes, and that was it. And that these guys would be less likely to fly at higher wind speeds. Okay, so here are our results, and to walk you through this, here you can see our fields where the measurements were taken. The color map corresponds to the model probability that you would find a tiny insect in that region. And over here we have emergence observations. This is the actual data. So here are the fields, and they're spaced proportional to the distance from the release. So they were released in field A, and G is the farthest away. And then on this axis, you have the number of days post-release. And this is our corresponding mathematical model. And so we used a Bayesian framework to come up with the best fit parameters. And in these near fields, the model does a pretty good job matching the data. But if we get out longer days in these farther away fields, you can see that quite a few insects show up in these sentinel fields, even though this is not in the direction of wind. And we were not able to capture that at all in the model. And so we felt that this made a pretty convincing case that in order to capture these not as frequent but definitely important long-distance dispersal events, that you really had to think more about the fluid dynamics. So you can imagine that there's a lot more complicated atmospheric flows than just a spatially uniform wind, and also some of the insect behavior in terms of detecting their hosts could also be important. So all of that was neglected, but our case here was to try to convince the ecologists we were working with that they should measure wind speed and they should think about behavior. And fortunately, we do have some collaborators now who are starting to measure wind speed when they release and collect parasitoids. So hopefully we will have better data to compare to. So just to summarize, if you're looking locally, you can do a good job with these simple models that describe the dispersal. At longer distances, you really have to think more about the details of the flow and the behavior. And then hopefully we can motivate people to think more about these questions. Okay, so a nice thing about this as well is that we could really see that these insects fly much farther than expected. And so now I want to shift from this population level to the aerodynamics of the spiders and insects themselves, which is, again, a complicated problem. So why is it hard to fly far when you're this tiny? One of the challenges is that maybe you can fly a centimeter a second, but you're really going to be driven predominantly by the background wind, even on days with a light wind. In addition to that, it becomes quite inefficient to generate lift to stay in the air as you get down to these Reynolds numbers. So where you end up is really driven by atmospheric flows, and then in addition, what you can do is quite inefficient, and you can't do it for very long. So all of these insects that I'm talking about live at this intermediate Reynolds number range of about 5 to 20. So this is the thrips with its bristly wings. I'll talk a little more about that. Other interesting creatures that live in this range are copepods and a lot of zooplankton. And for us, thinking about dragonflies and hawk moths, these are high Reynolds number organisms being around 1,000. So I'll show you that the aerodynamics is quite different for something like a thrips versus a dragonfly or even a fruit fly, actually. And we have done experimental work with organisms and also with physical models. But for the sake of time, I'm going to focus on what we've done with numerical simulations. And basically, our approach is to use the immerse boundary method to look at fluid structure interaction of a flexible dragline in the case of a spider or a flexible wing in the case of an insect. We're solving the full Navier-Stokes equations with this moving elastic boundary. And there's lots of interesting mathematics in terms of how to improve accuracy or speed. Since we moved to 3D and I do a lot of things other than writing code, we now use Boyce Griffith's immerse boundary method with adaptive mesh refinement. And this is freely available on GitHub. And the nice thing about it, if you have something like a jellyfish that is swimming in a fluid, your grid is adapted so that the finest grid moves with the organism and then also with interesting regions of flow like regions of vorticity. Okay, so let me just briefly highlight how the flow around the wing is different between these larger insects and tiny insects. What I'm first going to show you are vorticity contours around a three-dimensional simulation of a translating, or rotating rather, elliptical insect wing. This is at the Reynolds number of fruit flies. The angle of attack is 45 degrees. And these simulations were performed by Shannon Jones at UNC, and also with the help of Ty Hedrick and Arvind Santhanakrishan. And what you'll see here is what the flow looks like around most insects. So we have a stable region leading edge vortex and then a trailing edge vortex that separates. And the leading edge vortex remains attached up to high angles of attack, so you do not have to worry about stall for insects, and you can generate quite a bit of lift this way. Now for comparison, this is what the flow looks like for a fairy fly around the Reynolds number of 5. And you can see that the trailing edge vortex does not separate. Okay, so we have a stable attached leading and trailing edge vortex. So there are consequences for the lift and the drag that is generated. So the next graph I'm going to show you really uses the way that in insect biomechanics people have thought about comparing lift and drag over scale. And that is basically to report lift and drag coefficients using a high Reynolds number approximation that you take the lift or the drag force and divide it by density surface area velocity squared. Obviously, we're at Reynolds numbers 5 to 10, but the low Reynolds number approximation doesn't really apply either. So these lift and drag coefficients become functions of Reynolds numbers. You could think of it as a normalized force, but to make comparisons with the literature, this is how we have reported it. So here is a graph of that lift and drag coefficient across Reynolds numbers at a 45-degree angle of attack, which is about where Drosophila and these tiny insects fly. So the main story here, drag is in black, lift is in red. For Drosophila, dragonflies, hawk moths, and bigger insects, if you have this rotating wing at 45 degrees, lift and drag are pretty close. So you're generating about as much lift as drag. And then as you drop down in Reynolds numbers, you do see a drop in lift and then an increase in drag. So fairy flies that are about the smallest, you can see they're generating about 5 times as much drag as they are lift. And if your goal is to get up in the atmosphere because you're really going with the wind anyway, you have to generate quite a bit of drag just for a little bit of lift. And below these Reynolds numbers, just crudely between 1 and 10, you don't really see organisms that actively generate lift or thrust. Well, thrust for sure, but not lift. So again, this is about the smallest flying insect you'll see, and the spiders don't use active mechanisms. Okay, so is there anything that the insect is doing that can help enhance lift? And I'll show you some of our early results. So it's been known that all these insects clap their wings together and fling them apart, and that this can enhance lift. So the basic idea here is that you have an insect and their wings are horizontal. So they're not flapping like this, they're flapping like this. And so the wings clap together at the beginning of the downstroke, or at the end of the upstroke, beginning of the downstroke. And you can make a 2-dimensional approximation if you imagine just a slice along the chord, which is here. And then the wings begin to rotate apart about the trailing edge, which you can see here. And when this happens, 2 leading edge vortices form, and no trailing edge vortices form. And then as the wings begin to translate apart, trailing edge vortices begin to form and they grow, but for some time, the leading edge vortices are stronger than the trailing edge vortices, and this corresponds to greater lift. So in the next slide, I have some numerical simulations at a Reynolds number of 10. This is the lift per wing for 2 wings and 1 wing with the same motion. So you can see this first peak corresponds to the rotation of the wings apart. We get this nice peak in lift that is more than 1 wing with the same motion. And then this is the beginning of the translation apart. We see another peak, and then eventually, as the wings move farther and farther apart, and their influence of interaction decays, you would end up with the same lift. Okay, so this is a nice story. It does work better at lower Reynolds numbers. It works well at high Reynolds numbers, but the lower you go, the greater this effect. But there is a consequence to this, which is the drag that is generated. And these graphs correspond on top to the drag during the downstroke. So this very large peak is when the wings are initially accelerating apart. And over here, this is when they're clapped together, and this is the large forces generated to clap them together. So here is our peak drag during the initial acceleration, and this is the drag approaching what it would be during steady translation with 1 wing, and there's more than an order of magnitude difference. So you're getting about 30 to 50% more lift, but then you're getting over 1,000% more drag. And so you might think that this would help with thrust, but all this force is generated when the wings are clapped together and moving apart, so you have equal and opposite forces that cancel. So if these forces were generated later during the stroke, you would get some thrust, but it's not really helping with thrust either. Okay, so this, if you're looking at efficiency as lift over drag, is really not very good. So again, what are they doing? So you might have noticed that most of these tiny insects have bristles, and this came up in the talk on the pollen seeds earlier this week. So how can we think about what these bristles are doing? And you might think that, okay, if you're clapping together bristly, hairy wings at low Reynolds numbers, this might be easier. So the analogy I like to make is that you have these paddles. Maybe you've got ping pong paddles and corn syrup, and you're trying to smack them together and pull them apart, and it's very hard. And now instead you have tennis rackets, and maybe that's easier if there's some flow between the hairs. So all of these organisms are operating at this very interesting Reynolds number range that Cherry Cole, who's here, have looked at. So the thrips are about here, and what I'm plotting is the leakiness versus the spacing between the bristles. So if our wings were completely leaky, that would be a value of 1. If they acted like a solid plate, that would be a value of 0. These different lines correspond to different bristle-based Reynolds numbers, which for thrips is about 10 to the minus 2. The wing-based is about 10. But bristle-based Reynolds numbers were somewhere in here, and our spacing is somewhere around here. Okay, so if you have very small changes in Reynolds numbers by the wings moving faster, or maybe the right velocity is one wing moving relative to the other wing, so it's double the speed, you could be bumping your Reynolds number up and down in this nonlinear transition where the wings could behave very differently depending on the part of the stroke you're looking at. So looking at this numerically is quite challenging. We've got a whole lot of bristles, and because we're in this range, really resolving the flow between every bristle matters. So if you're not doing this well, if your grid isn't sufficiently fine, then you could end up with increased effective viscosity, and you're not accurately modeling the flow between the bristles. So my graduate student Shannon Jones did a very careful analysis in 2D, which basically we took a slice through the wing here and just modeled it as basically moving cylinders. And the grids that she used for this study, I think, were effectively about 4,000 by 4,000. But we did use adaptive mesh refinement. But very challenging 2D simulations, and we're going to try to go to 3D, and we'll see how that works. But right now I just have these 2D results. And I'm just going to show you the simplest case where we're basically looking at one row of cylinders translating versus 2 rows of cylinders moving apart. And I'm not going to rotate them, I'm just going to move them directly apart at a 90-degree angle of attack. So to walk you through this, the red arrows correspond to the velocity of these plates. So up here we have solid plates, 2 of them and 1 moving with the same motion. Down here we have our cylinders, and our cylinders are here, here. They're a little bit hard to see. So what happens in the case of the solid plate, so all the fluid has to move up and over and between the 2 plates as they move apart versus just one plate translating. And using the thrips morphology and Reynolds number, you can see that there is quite a bit of flow in between these cylinders. And here is the case of just the one row of cylinders translating. So let's see what the consequences for force are. We've got a range of Reynolds numbers, bristle base 10 to the minus 1, minus 2, and minus 3. This is log force, so the differences that you see are really quite big, bigger than they appear. And to walk you through this, the peaks correspond to when the plates are moving apart. So here they are during the acceleration as they move apart, and then that drag force decreases as they move farther and farther away. Dashed lines are 2 wings, so 2 wings are typically always bigger drag than 1 wing, moving with the same motion. But again, as they move farther and farther apart, those forces approach each other. And then the solid wings are this burgundy color, and the other colors correspond to increasing gap to diameter ratios, so they become leaky. And so the increase in drag, the first thing is Reynolds number dependent, going from minus 1 to minus 3. And again, this is log force. There is a very big difference between drag on 2 wings versus 1 wing. And then the effect of having these bristly wings It's also gap to diameter dependent, so you can decrease this large peak in force by having these bristly wings. So this green line is a little bit higher when you have two wings, but compare that to the solid wing. Okay, so the bristly wings, the bottom line is that when the wings are close together, for the thrips Reynolds numbers, they act like these leaky rakes, and as they move away from each other, they do act more like solid plates, and and that effect becomes more and more significant at low Reynolds numbers, and most of these very tiny insects do have the bristly wings. Okay, so whether or not this really helps in terms of lift generation, we're going to 3D with physical models to try to answer that question. So I want to just wrap up quickly by talking a little bit about what else they're doing, particularly the spiders. So I talked about active mechanisms, but there are also passive mechanisms of dispersal, especially in seeds, and also in these very tiny insects. So one thing that we noticed about the thrips in some of our recordings, and this is a little zoomed out, so we really can't resolve what the wings are doing, but we can see more of its flight. And after a couple of wing beats, it will spread its wings and just drift, and it looks very much like a dandelion seed. And if you think about how these guys are going hundreds of thousands of kilometers and they can only actively fly for about 30 minutes, probably a lot of their flight is just spreading their wings and floating on the wind. Now, of course, spiders go really quite far, and they really don't have a mechanism of active flight, although they can move their body and legs around to change their drag. But people, there had only been a couple of papers looking at the aerodynamics of spider ballooning and trying to think about how they could go these long distances that used sort of simple models of force balance, looking at the drag that the line might generate. And we wanted to test some other ideas to think about how they could go, again, hundreds of thousands of kilometers. And this is a group project that was started out of NIMBIOS in a collaboration workshop for women in math bio. So the basic idea of the models I'm going to show you quickly is that we have a flexible drag line. It looks pretty rigid here, but it is flexible in our simulations. And then there is a spider at the bottom that we make a point mass. So we are neglecting the drag on the spider's body, and we're going to incorporate that in the future. We're just looking at the drag generated by the silk line and then considering only the mass of the spider. And for the Reynolds number range, if you base it on the length of the drag line, it goes from 100 up to 10 to the fifth, actually. If you base it on the diameter of the spider's body, somewhere between 10 to the minus 1 to 10 to the third. And so this does end up looking like a high Reynolds number problem when you think about the length scale of the drag line. This spider on your left, so here is the drag line. There's a point mass at the bottom. We just started these at the top of the domain and let them drop. And we're increasing the mass, which effectively increases the Reynolds number because they fall faster. And up around where you would find most of these spiders, you can see that there is a nice wake of alternately shedding vortices. And if we look at the set length velocity versus the mass, we end up with a square root relationship. So set length velocity scales with the square root of gravitational force. So drag scales like the square of velocity. So again, high Reynolds number. And set length velocity is indirectly and linearly proportional to the drag line length. So none of this is perhaps too unexpected, but it was nice to validate just quickly with some CFD. And so we release these spiders and they always fall, and sometimes they fall slower. So how are they getting up to 5 kilometers? We did some studies where we had updrafts, and so we have upward wind at 30, 45, and 60 degrees from horizontal. And if it's a really upward wind, you can get the spider to take off. But For it to live in a sustained upward wind for thousands of kilometers seems unrealistic. This might help them get up into the atmosphere a bit, but how are they maintaining that flight? There was an idea that they could be transported in eddies, so we did a couple of simulations. This is a lid-driven flow just to generate some vorticity. The spider is tethered and then we release it. And the spider sort of goes around and around. So fun simulations to do for a workshop. I don't know that a spider would actually experience this, but one of the things that we found is that the spiders do go round and round, but on average they keep falling down and down. So the net flow down is the same as the net flow up. So we weren't able to capture this idea that you could have sustained transport if the spider was well positioned within an eddy such that the mass downward was balanced by this upward velocity. So we weren't able to capture that. And we started thinking a little bit about what else was going on. So again, with these quick studies, we found, all right, it's a high Reynolds number problem. You have to have this constant upward wind to keep the spiders from falling down. And so the last finale, since I'm out of time, is that we decided to look at density layers, particularly since these guys are found in density stratifications due to temperature inversions. I'll just say we took our little spider, we put it in a Busenek's approximation for the fluid, and we dropped the spiders again. This is a lighter fluid on top and a heavier fluid on the bottom. The spider is heavier than both, and we drop them. Whoops. Let's see. There we go. Okay. And what you'll find is as it goes through this density stratification that it can get stuck there for a while. And with this very long drag line, you can imagine that it's coating itself in lighter fluid. It hits the denser fluid, and effectively the spider plus boundary layer is lighter than the heavier fluid on the bottom. And so you could have these long residence times due to these density stratifications, with, in addition to some upward flow, might allow them to stay up in the atmosphere for a long time. All right, so it's still a difficult question. How can these tiny insects travel hundreds to thousands of kilometers? They can't actively fly for more than 30 minutes. They generate a ton of drag and very little lift. They don't fly in hurricanes where you could really imagine they go very far. Again, it's on very light, breezy days. So we need more fluid dynamics. All right, and to summarize, many people to thank and funding agencies. So happy to take a couple questions.
Maybe while the next speaker comes up to set up, we have time for quick questions.
Are there estimates of the bending rigidity of the threads?
Yeah, and we put those in. And so they're very, very flexible. And the silk threads are charged. So I think that they're getting some additional stiffness, or at least not getting tangled up through electrostatic repulsion. So that's another aspect we want to look at.
Okay, I think for the interest of time, we'll take the questions offline, but let's thank Laura again.